Hi class, Dr. Jim here. In this lecture, we're going to be looking at molecular biology. So I did this one a little bit different than all the other lectures that are coming straight from the book. This one does have a lot of the information that is from the book, but I've kind of added a few touches since a lot of my research was looking at some molecular biology, and these were some of the techniques that I would do in the lab, as well as some of the other ones that we'd use in research that are kind of cutting edge, or were cutting edge when I was going to school, and now kind of the basic everyday type of thing that we see in a biology lab. Now, what's interesting is that the reason why I think this chapter is so important for you guys, especially going into medicine, is medicine is becoming much more personalized these days. This means that instead of looking at a big group of patients, we're really boiling it down to the single patient and seeing what treatment can be the most effective at treating things. This especially is taking place in oncology, looking at cancers, treatment of cancers using the immune system, and talking about specific receptors and T cells and how we can fight those different types of things. We see this with infectious disease, where again, a lot of times instead of using antibiotics, using other techniques to maybe minimize antibiotic resistance and attacking it either using the immune system or some other method, maybe like a bacteriophage in those cases there. Or finally, in transplant medicine, where again, before it was we had to find a suitable donor. But in the future, hopefully, if everything works the way that we hope it does, is that one day that you may go in and actually build your organ that you may need to transplant yourself by taking stem cells from your body and building that organ, and then very shortly after, once that organ has been built, you now can put that in and exchange it for something that was not effective. And so a lot of this seems science fiction, but a lot of this is actually coming very quickly, and we're seeing a rapid change in the way medicine is approached. So this is, I think, a really good chapter to kind of go through some of these different techniques, some of the basic techniques that any molecular biologist would know, and then kind of getting into these future studies of where these things are and what's actually being done there. So that's kind of why I like this chapter and taking you through some of these different things and showing you guys where it started and where is it going. And I think that's going to be important today. Okay, so let's take a look at some of the different aspects of molecular biology. And first, looking at really the main heart of molecular biology, which is the DNA, and then building everything off of that. So one of the interesting things about DNA is that DNA is a double-stranded helix. Now, we've talked about this before in chapter 2 when we looked at types of bonds. And one of the interesting things is that the bond between the bases, and that's the A's, the T's, the G's, and C's, have these things called hydrogen bonds. Now, hydrogen bonds are weak bonds. They hold the bases together really well. However, they're very weak in the sense that you don't need a lot of energy to put into them to break those bonds. And in fact, all you have to do is actually heat them up, and you can break those bonds pretty easily. So you don't need really an enzyme to do that. You just need some heat. Now, in our bodies, when we're doing replication or doing transcription translation, we can open and close the DNA fairly easily using certain enzymes like helicase or RNA polymerase or something else that actually unwinds and opens the DNA to do that type of thing. But in the lab, we don't necessarily have those enzymes to actually work, and so heat is the next best option for these things. And so what you can do is if you apply heat with those hydrogen bonds, you can actually open them up. And again, we've talked about this before in chapter 2 and then also in chapter 9, looking at the different types of hydrogen bonds. And again, remember, these are weak interactions. They hold these things together pretty well, but they're also weak in the sense that you don't need a lot of energy to break these things apart, and that's going to be the key. We can heat the DNA up to 95, and that will separate the DNA out into single strands, which is a good thing because then we can get at the bases and look at a number of different things that way. And then what happens is if you cool those strands back down again, they'll find their binding partner and go back together, which is kind of a nice little thing. So just simple heating and cooling allows us to open and close the DNA, which is really important to a lot of the techniques that I'm going to talk about today in molecular biology, and that's going to be really important. So I want you to kind of keep that in the back of your mind. Now, I've got a nice little graphic to show you here of what actually happens when you heat and cool the DNA. So let's say we have a strand of DNA here, and again, it could be any of these base pairs. I just kind of put it color-coded so you can kind of see the complementary base pairing in that and the hydrogen bonds between them, and that's indicated by those white little lines. Now, if we heat the DNA, we can separate the DNA apart. What happens is the heat breaks those hydrogen bonds break apart, and now you get two single-stranded pieces of DNA. Now, if I leave those two pieces of DNA in that same container, typically in a little microfuge tube, what I can do is then just cool it back down, and the DNA will find its binding partner and go back together once that cools down again and form those new hydrogen bonds. So that's a great technique to have, that you can just heat and cool that DNA and open and close it. And what's also nice is that you don't ruin the integrity of the DNA. The DNA is very stable, and that you don't see the individual bases actually breaking apart off the DNA. So that's really helpful in some of these techniques like sequencing and PCR, where we have to reheat the DNA over and over again to get these strands to separate and allow us to build new strands. And so that's one of these critical things, is that DNA is pretty heat tolerable in the sense that the normal phosphate, base, and sugar are very strong with just simple heating. It doesn't break it apart and degrade it very easily. And so that's one of those things that's really nice. All you're breaking is those hydrogen bonds and separating out those different sequences. And so this will be very important. Like I mentioned, a couple techniques that we're going to talk about here in a little while. But I just want you to be aware of why molecular biology works in the sense. And really, this is kind of that first principle that everything else is based off of. And we talk about these things today. Okay, so just like a craftsman has a toolbox to, build furniture or to work on projects at home, or if you like painting or sewing, everyone has something like a toolbox where they have their go-to tools in order to do these things. It's no different in biology, and especially molecular biology. When I was working in the lab, my advisor would always say, hey, you need to have a toolbox of these different things, and if you don't know the technique, go ahead and learn them, because these are going to be important and they build on one another. And so getting that experience and learning those things is very important. What's interesting now is a lot of these techniques that were, new and exciting when I was in graduate school back 20, 30 years ago are now very basic techniques that we're teaching a lot of people in middle school and high school using these wonderful kits from these different biotech companies. So it is something that's kind of working its way down and now becoming available to almost anyone that wants to learn these things. So again, these things are important, and I'll talk a little bit about each one of these today, but this is what makes these future experiments and future studies very exciting, because we're using the techniques that we learned in the past to do bigger and better things in the future. And so let's take a look at some of these different things in the molecular biology toolbox. So the first thing we need to know is how to cut DNA. So we need a pair of scissors. And so I'm going to show you some enzymes that are out there that are very useful in going in and cutting DNA. And so that's the scissor part of cutting DNA. And so that's one thing you want in your toolbox. The second thing is, well, if you cut DNA, you have to have a way to put it back together. And so that's going to be important. You just don't want to have these random fragments just scattered about. You want to be able to glue them back together and mean something. And so that's going to be important. And so I'll show you the particular enzyme that we use in molecular biology to glue this DNA back together. Another important aspect is being able to measure the DNA. So when you cut the DNA, a lot of times you make these fragments, and you need to understand what these fragments are and a way to kind of either measure them to figure out what do you have, and then the other thing is, can I use that to then make bigger and better things? And so that's going to be important as well. And so not only does it work really well in finding out little fragments, but it can tell us a lot in bases that are shared between organisms. We look at a lot of times even paternity tests and DNA. So like if you've done the 23andMe, a lot of these things are used by using this measurement technique, as well as taking pieces from one organism and putting it into another. Very important. And again, this is a basic technique that is done all the time now using this gel electrophoresis, and we'll talk more about that in a few minutes. Again, copying DNA, a lot of times, if we're talking about one individual cell, even though there's a lot of DNA in those cells, when we talked about this in chapter 9, that it stretches out for 6 feet, you need to be able to work on specific genes and gene targets. And so in order to do that, you need more than just one piece of DNA. You need lots of DNA to do these things. It also helps with forensic biology, helps with molecular biology, and a number of other techniques that we use. We need to be able to amplify that DNA so it makes it able to be worked on. And so again, we'll talk a little bit about copying the DNA, and then finally understanding the DNA code. And so this is really what's changed medicine in the last 20 years, is being able to sequence the DNA and say, what do these A, G, T, C's all mean, and what did they tell us about our genes? What's in the genes, what's there, what's not, how can we change those, edit those types of things. That's going to be all that new technology that we're going to be talking about, and we'll look at some of those things as well. But if we didn't have a way to read it, we wouldn't be able to know what the sequence was. And so that's what's nice about this technique. Again, it's another technique that was from the late 70s, early 80s that allowed us now to really get a hold and an idea of what the genes are and what the genes actually make, which is the proteins. And so that's going to be important as well. Okay? So the first thing we're going to look at is the restriction enzymes. Like I said, we need to be able to cut DNA. And this is important because we want to be able to, take genes from other organisms and pop them into these things. Maybe one organism has a really important gene that we'd like to take and put into another organism, or maybe make a product out of it. And so in order to do that, we need to be able to cut the DNA. And so that's going to be really important. Now, these enzymes were discovered a long time ago when we were looking at different species of bacteria. Some fungi have these things as well, but a majority of these things we discovered in bacteria. And these are actually natural defenses. So I talked about exoenzymes before in Chapter 8 and releasing these enzymes out to, either obtain material or a way to fight other bacteria, other species that you're going against. We talked about that microbial antagonism before. So these enzymes are actually really important for not only cutting up DNA, but cutting up your competition. So if you release these enzymes out there and you put them on your competition, they get uptake by your competition and they start cutting up their DNA. Well, if they don't have their DNA as solid fragments anymore, or now broken up into fragments, they're not going to survive very long. So it's a way to get rid of your competition. So what scientists have done is looked at this mechanism and said, hey, we can actually use these enzymes to actually cut the DNA in other organisms, and then be able to take these pieces out and maybe use them either as a way to harvest genes out of one organism and put them into another, or maybe to get these individual fragments so we can look at genomes or other things like that. So that was really the initial idea, is using these enzymes as a way to cut the DNA so that we could actually see fragments and be able to use that DNA in the lab. So to give you an example, this is one of the first sequences that were actually discovered to cut DNA, and this is the EcoR1. Now this is an enzyme that E. coli produces, so that's where the eco comes from, and R is another R1 with E. coli. And then this 1 is restriction enzyme 1. And so essentially it just means this is the first enzyme they found in E. coli that actually could cut DNA. And what they were able to do is figure out the exact sequence that this enzyme targets to cut this DNA. And typically when we talk about these enzymes, they look for a specific piece of DNA. And what you notice here is if you look at this GAA-TTC is a palindrome of itself of GAA-TTC. And that's what these enzymes look for, are palindromes in the DNA sequence. And when they find that palindrome, they bind to it and then they excise the DNA. So when I draw this little line here, what that represents is where that enzyme actually physically breaks the DNA into the different fragments, and that's going to be important. So what happens is you cut that fragment like so, it separates out, and now you have these two fragments. Now I want you to remember, you see those little flaps on there? That's going to allow us to glue to other things. So it's going to be important down the road when you think about it. But just kind of think about these little flappy things. We call these sticky ends because they're flappy and they can stick to things. So I want you to kind of remember that when we use these things in the future, when we glue these back together. So that's one of the things that we talk about here. Okay, so let's say in our sequence here we see if we have one of those things, and sure enough we have a GAA-TTC. Here's the GAA-TTC. So that EcoR1 enzyme would come in, it would cut that DNA, and now it would slice it and produce a different fragment. And so now you've cut the DNA in one spot. Well, like I mentioned, there are many different enzymes out there by many different organisms, and there are actually biotech companies that sell these readily to the local scientists out there. So if I was working in a lab, I can call up New England Biolabs and say, hey, do you have this EcoR1 enzyme? They say, sure, send it to me on dry ice. I get it the next day and I'm ready to go, and I can start slicing some DNA up. And basically they have this huge long catalog of all the different enzymes that you can look up from a number of different species of bacteria. So let's get another example of another piece of DNA. This is from Haemophilus influenzae, and this is the third restriction enzyme sequence that they found. So here's the sequence. Again, it cuts the DNA, and again, you notice they have these flaps on them. And so again, allows them to stick to things. And we'll talk about more when we start gluing the DNA back together. Well, essentially, again, if you have that sequence, you can cut the DNA, and now you've created 3 fragments. So these enzymes are really important for allowing us to cut specific sequences out of DNA that we may want to use for something else. So let's say taking a gene, and we know that there's some sequences on either side that we can cut the DNA out and pop this gene into another organism. Or maybe if we're looking at how closely related one organism or one species is to another, we can treat each genome with enzymes and take a look at these things. And again, I'll show you how we do that down the road here in a few minutes, but that's another thing that you could potentially use these enzymes for. So lots of applications for these enzymes, and I want you to get this idea that this is one of the reasons why we can do all the things we can do. Without these restriction enzymes, there would be no way to cut DNA as easy as we can in the lab as we do today. So that's the first thing, okay? The second thing is, well, now that we cut the DNA, we need to have a way to glue this back together. And one of the enzymes we use is a natural bacterial enzyme again, which is a ligase. And a ligase is another enzyme that allows us to glue the DNA back together. And so what we can do, as long as the DNA fragments match one another and they're complements of one another, we can glue these back together. So you notice here I have a sticky end here and a sticky end here, and they complement one another. I can put a piece of glue or drop a piece of glue in there and glue it back together. Now that works very nicely. Nice sticky end sticks it back together. Same thing here. I've got complementary sequences here. I can put a dab of glue here, right there, and glue it back together. Think of it like if you broke your mom's favorite vase and now you have to put all the pieces and glue them back together. It's much easier when you have little ridges that stick out. What's hard is when you have end-to-end. So a lot of times you'll have these enzymes that cut right down the middle. And if they cut right down the middle, we have what they're called blunt ends. And those can work, they're just much harder to stick back together. It's kind of like in woodworking, you never want to really try and glue anything together. It's not a very strong joint. What you always want to do is try and find something that overlaps. And when you overlap something, it makes a much stronger joint and it doesn't come apart. Whereas if you put it together like this, all you need is enough little bit and it breaks apart. And so that's the issue with these blunt ends. A little bit harder to work with, but there are some that do cut just like with the blunt end, just like so. So that's going to be important there. Okay, so just think about it that there are enzymes out there that can cut the DNA. There's enzymes out there that glue it back together, and now you can put fragments back together however you want. So why is this all important? So again, I kind of brought up this scenario. We can take genes from one organism to another and pop them in and do these things and do all this other fun stuff with them. Well, this is important in the idea of making proteins. And so again, DNA codes for proteins, doing transcription, translation. And so what you need to do is find a gene that you want that makes a protein that you want, and then find a suitable organism in which you can make that protein in. And a lot of this has been done in E. coli because E. coli, we can use E. coli very well to take up plasmids and put DNA in, and that's what I'm going to show you next, is how this works in a practical application. So let's say we have a bacterial DNA, and now we want to pop in something that would be very useful as a protein product that we could sell as potentially as a pharmaceutical. So this was the idea back in the 1970s, is that before this time, when people needed insulin, they had to go and basically use porcine insulin, which is basically pig insulin. And the way that they got this was a hard, laborious type of process where they had to take pig pancreases from the slaughterhouse and purify porcine insulin out of them, and that's how you got it. And so it worked, and it worked pretty well, but it was a hard process to get the porcine out. Plus, taking it from another species always brings the idea of potentially getting allergic reactions to it and causing a lot of problems. Wouldn't it be nice if we could actually use human insulin and be able to make lots of it very quickly? And that was the idea back in the 70s. And so we have the system, we can cut DNA, we can glue it back together. How about we take human insulin, take the human gene, pop it into bacteria and see if we can make human insulin. So the idea was, okay, we have this bacterial DNA, we'll cut it with an enzyme, Okay, just a simple enzyme. You can see we have these sticky ends. Then what we'll do is we'll take the human insulin gene, we'll glue it into the bacterial pieces, just like so. Remember, you have those sticky ends, now you can glue them back together. We pop that into E. coli, and then all of a sudden, now we let our E. coli turn into little factories, and they pump out this protein, which we can purify out of the E. coli very easily. And then if you do that, what happens is you make insulin. Now you make what we call this recombinant insulin. You can give that to people. You make lots of money off of that pharmaceutical product, and you become a big biotech company. And that's basically what happened in the late 1970s when they were able to manipulate this system and make human insulin gene. And so that was just one of the first products that they do. And in this next slide, I'm going to show you a number of different recombinant products that are out there today, including a lot of immune treatments like interferons, interleukins, which are all proteins that are necessary for turning on our immune systems. Macrophage colony stimulating factor, which basically stimulates macrophages. Tumor necrosis factor, which is another cytokine, which is used, and then granulocyte colony stimulating factor. So all these proteins can be given to a patient who is suffering a major infection and help ramp up their immune system so that they can fight off this infection. So that's something that's really important that can be done very easily. Other things like enzymes, so antitrypsin, this SOD, which is superoxide dismutase, and then lipases, which are used to break down lipids and that stuff, are other enzymes that are useful, again, in human conditions where maybe you don't make the protein or you have insufficient proteins. And then hormones like insulin, we talked about that already, erythropoietin, tissue plasma activator, which is basically blood clot, or with blood clotting to break up blood clots. So it's typically used after someone comes in with the conditions of a stroke, and so breaking up those clots, or maybe an embolism or something like that. Hemoglobin A for those that, again, are not making typical hemoglobin, so again, having issues. Factor VIII, which is a blood clotting, so maybe you have hemophilia, you need that protein. And then we've seen a lot with human growth hormone with HGH, where people use that to, especially with dwarfism, or, where you have issues where the body doesn't develop normally, you can use this as a hormone to develop it to a normal size again, develop the body to a normal size. So those are just some of the things that are out there that are actually made in E. coli now. They're called recombinant proteins. So you may be taking one of these things. I don't know, if you're taking one of these things as a treatment, this is possibly something that you may see in your future, especially if you're, delivering the medicine or being a patient yourself in these cases there. And you can see, and this is just touching the iceberg, the tip of the iceberg. There are many other proteins out there that are being used in the same way as a treatment there. And this is just a small list of what's just out there, doing it the same way that I talked about with human insulin, popping it in E. coli and allowing for that protein product to make. So kind of interesting in seeing those different things there. Okay, the next thing we want to do is actually, now that we can make fragments using restriction enzymes, is a way to figure out what those fragments are. And so one of the ways that we can do this is by basically measuring the DNA, putting it on something and using it to figure out what are these fragment sizes, what do they tell us about the organism, those types of things there, and we can use it to our advantage. And one of the ways that we can do this is a process called gel electrophoresis. And essentially, this process was found back in the late 1950s, early 60s, where they started using agarose as a way to run DNA down these things. So they knew what DNA was, but using it as a way to do these things. And again, it's using basic principles of DNA that allow for these things to happen. So how does the system actually work? Well, if you've ever seen the game Plinko on Price is Right, this is kind of the idea behind it. So you put your DNA at the top where that Plinko board is, and then you let it run down that gel in order to get to the bottom. Now, what happens is that we work on it based on size, because little pieces can move through those pegs very quickly and large pieces get caught. And that's the whole idea behind that idea. Now, we don't win big money if we run our DNA down a gel like you would on Plinko, but it works pretty much the same way. The idea is that you have your DNA at the top, and then you run it down through the little pegs, and like I said, the small pieces get through much quicker, where the larger pieces get hung up. So what it does is it allows us to separate DNA based on size, and that's going to be the key when we look at these things. Small pieces are going to move faster, they don't get hung up, and they can move much faster through the agarose gel. So think of those little agarose beads as the same thing as those little pegs in the board, and those little pieces can move much faster through, where the longer pieces kind of get tied up and move much slower through the gel. And that's how this process works. Now in the lab, this is kind of the setup here, showing you the gel box and how this thing works. Now we don't use gravity like we do in the Plinko board. Basically you drop the chip down and it works its way through gravity. What we use here is basic chemistry principles of DNA. DNA is a negatively charged molecule, and we can basically attract DNA by having a positively charged pole. Remember, opposites attract. And so if we apply electricity to that gel, what we can do is move the DNA down the gel based on its size, and it separates out based on its size, all moving to this positively charged pole. Now let's say we flip the electrodes and flip it upside down and put the positive side up here. Well, then the DNA is going to run in this direction, and it's going to run right off the gel, and you're not going to be able to see anything. So it's important that we make sure that we run it correctly in the right direction. And so again, that's going to be important. And they made this pretty much foolproof because they basically give you two colors. The red is the positive, the black is the negative. You plug them into the right port, and you still run the gel, and it should run properly down. Now the other interesting thing is we use like this loading buffer. I don't know if you can see it very well here. In this gel, you can see this loading buffer right in here that's kind of a bluish color. That allows for the DNA to sink into the well and then get caught and run into the gel itself. And in this, a lot of times we'll use these stains that actually stain the DNA so that when we use a UV light, we can see it. And this is under a UV light. This is using ethylene, or not ethylene, but ethidium bromide that allows it to light up when you apply UV light to it. There's other techniques now that just use things like methylene blue and other things that are much safer. The problem with this, with ethidium bromide, is it likes to stick to DNA, and so it could be potentially carcinogenic if you use it a lot. So someone that works in the lab, we want to try and reduce as much carcinogens that we're exposed to, and so trying to get nicer, easier ways, especially when you're working with high school or middle school students running these things, we want to make it as safe as possible. So you may see this sometimes used in certain labs, but a lot of times now we use the very easy methylene blue to stain the DNA, and it allows us to see the DNA pretty well. Now, like I mentioned, you get these fragments out of here, and they're all based on the different sizes. The smaller fragments down here are smaller, and they're going to run faster, while these larger pieces are going to run slower, because they get tied up. Now, a lot of times what we'll do is we'll run what we call a molecular ladder, and this is a molecular ladder here. These are known size pieces of DNA, so when they run down the gel, we can use this to our advantage, and we can build simple graphs to kind of basically say, okay, if we run this far, we can measure with a ruler how far down it is and give us an approximate size for all these different fragments. So this really bright band here is typically about 1500 kb. So we know that anything above this 1500 right here is going to be bigger than 1500, and then all these other ones may be a different size. And they have a number of different markers out there, but that's just some of those things that are out there to give you different sizes. Where these down here are around 500 base pairs, because these are about the 500 base pair pieces, and this would give you an idea that they're around 500. So kind of give you an idea of how these ladders work and that stuff. You can also play around with the concentration of the agarose, and that the more agarose you have, the more beads get put in, and so the more separation you're going to get out of the smaller pieces versus the large. And so again, there's a lot of different ways to play with it, but I just wanted to kind of give you the basics today of how it works. Okay, so what can we use this for? Well, one, we can use it for is comparison of different organisms. So if we're an evolutionary biologist and we want to look at the DNA, which is really important now, looking at species to species and looking at their DNA to see how closely related are, we can cut the DNA and see where are genes shared. And so we can see that there's fragments here that are shared between all three of the organisms, and then there are other fragments out here that are not. And so we can look at those and see, based on sharing, that we could say that, this is kind of interesting, that maybe organism 1 is related to organism 2, but if you look at organism 1 to organism 3 or 4, they're not as closely related as the 2. And vice versa, organism 3 and 4 look more closely related than maybe 2 is to 3. And so again, you can go back and forth and look at some of these things, and just by looking at the fragments, we can see who are more closely related to one another based on the sharing of those different fragments. And so that's one of the ways that evolutionary biologists are actually using molecular techniques to identify species and how closely related to one another. The other one that we see it a lot these days is in simple tests like paternity tests that are out there. And again, I know that everyone's out there has seen a Maury Povich episode of Who's the Daddy? Well, this is essentially what they do. They basically take DNA from mom, DNA from baby, and then the potential fathers that are out there, and they look for, again, shared fragments of DNA, and those fragments that are shared between the child and the father means, aha, you are the father, you are going to be the one that's paying the child support. And you can see here, Bob shares one piece of DNA, but Larry shares 3. And so that makes sense. And so Larry, you are the father. He's going to be the one that's going to be paying the child support. And so he's the one because, again, the matching sequences. So there are a lot of potential uses for doing this gel electrophoresis. This is just one example using paternity, but we've seen it using like 23andMe and a number of others, Ancestry.com and all these other things that are out there, and seeing, these different types of things that are used for identification of different species, and then also how related inside species these organisms are. So very interesting indeed when we look at these things. Now the next step is being able to have enough DNA in order to actually use it to cut and do these other things. In order to do that, we use something which is called PCR. PCR stands for polymerase chain reaction, which is basically the setup of how this actually works. We use this to make millions of copies of DNA so that we actually have usable amounts of DNA in order to cut it, to sequence it, to do all these different things. And so really, this is pretty much the first step. If you don't have enough DNA to work with, you're going to go to PCR first to make all your DNA, and then you can go back and then cut it, sequence it, do whatever you need to do in order to see what you need to see. And so this is basically using the principle of DNA to open and close, like I talked about at the beginning. We use that to our advantage. We open the DNA and close it back up so that we can make more fragments, and it allows us to work. Now, in order for this all to work, one of the issues is we use normal DNA polymerase that's taken from us or bacteria like E. coli. Unfortunately, those polymerases are not heat stable. They're going to fall apart if you heat up the solution. Those proteins become unfolded. And again, that's kind of what we talked about in Chapter 8 when enzymes fall apart. They get denatured due to heat. Well, one of the enzymes that we can actually use is taken from an archaebacteria that lives in these wonderful hot springs. I mentioned these before with the archaebacteria. These archaebacteria can tolerate lots and lots of heat, so their enzymes are very heat stable. And so one of the ones that we use is this Thermus aquaticus archaebacteria that has this wonderful thing called Taq polymerase. And this Taq polymerase, which is named after Thermus aquaticus, you can see the T and the AQ here, is basically the polymerase we use to replicate DNA. And so it works really well in making lots and lots of copies because we can heat up this enzyme, it's heat stable, and it's not going to fall apart of us if we actually heat it up to 95 degrees and then cool it back down again. And we can use it over and over again without worrying about losing the integrity of this Taq polymerase. And so that's going to be one of those interesting things about it. So how does PCR work? So let's go back to our sequence that we talked about before. Here's our wonderful sequence, and what we need to do is add some nucleotides because, again, in order to make more copies of DNA, you need nucleotides to build those things up. Mentioned you need the Taq polymerase from that Thermus aquaticus to have something that's heat stable that can be used to make many fragments after heating and cooling the DNA over and over again. And then you need these primers, and the primers are going to be specific to the DNA sequence. So typically you know the sequence that you want to replicate over and over again, so you can build these primers accordingly. So a lot of times scientists will build primers in the lab using computer programs. They send it off to a company. They make the 20-piece oligonucleotide, they call it. You get it back in about 3 days, and then you can be on your way making these things. So if you know the sequence already, you can build exact primers that are going to amplify the DNA. But in some cases, you may not know, so you may use something that is like kind of just to amplify DNA or to get something that's close to somewhere that you want to amplify. And so again, kind of knowing that sequence does help, But there are ways around it if you don't know the sequence, so you can amplify the DNA. And like I'm talking about, like with human DNA for forensics and that stuff, there are certain primers that can be used in human genomes that allow it to amplify certain pieces of DNA, especially those repeats that we have. And I'm not going to get into that today, but that's one of the ways that we can actually, if we don't know the sequence, how we can replicate the DNA very easily. So these primers are going to be real important. I'll show you that in a minute, along with the TAC and the nucleotides to actually do the PCR. So let's start this in motion and see what happens. So what we're going to do is start our cycle. We heat the DNA to 95 degrees. We then cool the DNA down to allow for these primers to anneal to the DNA sequence. We then heat up the DNA to 72 degrees that allows the TAC to actually run. And so we have these 3 steps in the cycle, which is basically heat, cool, and then extend. Heat, cool, and extend to allow for the TAC polymerase to do its job, take those nucleotides, and make 2 new fragments. So after 1 cycle, we've now made 2 fragments. So let's say we started with 1 fragment of DNA. We've now made 2 in 1 cycle. If we repeat that cycle again, a heating and cooling, we go through, we now make 4. Then we can go back and do another heat cool, make 8. And you can see as we continue on and do up to 30 cycles of PCR, we can make over 1 billion copies of DNA. Now, normally we stop at about 20 to 25 cycles because having 1 million pieces of DNA is probably enough. But if you really want to be sure, you can go up to 30 cycles and do it. Now, like I said, most protocols call for anywhere between 20 and 25 cycles, and I think that gives you a fair amount that you can see on a gel and you can use. If you're doing something where you want to take that DNA and put it in and want to make sure that the sequence is perfectly correct and not making any errors, there's these things called high fidelity TAC, which are going to make sure that it proofreads the DNA and everything else. But typically, when we do this, we're just interested in making copies. We're not so interested in, if we add a different letter here or there just to get the fragment that we want. That's perfectly fine, and that works really well. And this only takes a matter of hours, or a couple of hours, and you've now had millions to billions of sequences of DNA. Much of the DNA that we can use to, again, manipulate by cutting, gluing in, and then even reading, sequencing, and doing these other things that we want to do to it. So very useful process that's out there. So why is this important? Well, again, we talked about forensics before. Taking a small sample of blood or other things that are out there, we can use this in forensic laboratories to replicate the DNA and see crime scenes, like who's left their DNA behind at the crime scenes, identification of maybe someone who, where you can't identify it by other ways. Maybe you can do DNA forensics on it and identify the victim in these cases, or some other mechanisms that's out there. Or, like I said, in general molecular biology, maybe you're working with a specific gene. You want to amplify that gene so you can put it into E. coli and make protein out of it. This is a great way to do it by doing PCR, figuring out what the gene, so you get plenty of copies that now you can glue into the genome, or into the gene or the cloning vector that allows you to do that. And that's going to be really important there. Now, the last thing, like I mentioned, that's really important in molecular biology is sequencing. And so this has really changed a lot of what we know about genes and genomes and all that other stuff in the last 20 to 30 years as a method of being able to read the DNA. And so when we first did this, it was a very difficult process and a very slow process that in order to sequence a gene may take you a couple months to a year to actually do. Now we've automated this process. It is very easy, and now we're doing entire genomes very, very quickly. And I'll talk about that in a minute, especially with the human genome that we now have and we can use lots of copies for. So in order for this mechanism to happen, it's a lot like PCR, where we're going to open up the DNA, we're going to use normal nucleotides here, and then we use these special nucleotides that have these colors to them. So this is why I changed the colors so that you can kind of appreciate the colors. And they're called D-deoxy because essentially once you put this nucleotide in, what happens is it basically stops the sequence from being built any farther. So it kind of has a stop sign at the end and says, this is as big as the sequences that you're going to make. So when this process takes place, the DNA polymerase has a choice to make. It either uses the nucleotide here, or it picks one of these special nucleotides that are in the mix. And depending on which one it makes, or which one it uses, depends on how big the fragment's going to be. And I'll show you that in just a minute. The other thing we use is simple DNA polymerase, because we only really heat the DNA once to open it up, and then that DNA polymerase is stable enough for us to do sequencing with it and doing this pretty well. So again, this is a process that doesn't take very long. We add the DNA polymerase. The DNA polymerase goes along with the DNA, like so. Again, you're going to use primers like you had before to build the DNA, and what you do is you start building fragments. So again, those fragments get built. Now remember, normally DNA polymerase will just grab normal nucleotides, but eventually what will happen, it will grab one of those D-deoxynucleotides, and what happens is that they stop. And when they stop, they can't build a bigger fragment. And so you can see that if you continue to do this, it will build bigger or smaller and smaller fragments of DNA based on where they put those nucleotides. And so eventually what happens is you go all the way down to the end. Now, I didn't have enough room to put all of them in there, but eventually what you're going to make is a fragment of every size. And basically the fragment of every size correlates to the gene sequence. And so the other interesting thing is, like I said, is these things are labeled with a fluorescent dye. And so now, instead of using our eyeballs to be able to see the fragments, which is done before, now we can use computers with lasers to actually be able to see these nucleotides and give us the sequence very quickly. So what happens is you take those fragments and you run them on a sequencing gel like so. So again, remember those colors. You run them on those gels, and those gels would tell us the sequence based on, again, small fragments on the bottom would be the smaller sequence, and then as you work your way up, you can build that sequence up. Nowadays, and so that was the hard part. You'd have to run these gels, you run the sequence would take a while to run, 3 or 4 hours, you'd run the gels, and then it may take you a while to read it because you're going in and looking at each individual base pair, trying to figure out how they line up. And again, depending on how well that gel ran, it may be really good or it may not be so good. And so depending on how well you ran those things, you may get really good sequence, you may not. Nowadays, being all automated, now what happens is they don't really even run a gel anymore. What they do is run these little graphs for the computer to read, and the computer can read based on where they find these different sequences. They can see the color and put in the approximate letter that you need, and so it builds the sequence very easily, and then you get out this thing. So this process has become totally automated, and essentially what we have is a technician who basically puts in your sequence that you want, loads the nucleotides, loads the D-deoxynucleotides, the DNA polymerase as a big mix. You mix that together with your DNA, you let the computer run it, they screen the DNA for the colors, and lo and behold, they give you a sequence. Nice, beautiful sequence every time that you can read, and it reads very nicely, and then you can use that as a way to say, okay, I've sequenced this, I can do this. And now most biotech labs, most schools, big schools that have, cloning labs and everything else, have their own sequencer that basically they can go in if they need to find out the sequence of a fragment that they don't know that they have. They can quickly do it within a matter of hours, have a complete sequence, and then be able to say, okay, we can build primers, we can now do PCR, we can do a lot of different things with that and be able to do these things very, very quickly. So why is this important? Like I said, one, it's be able to see individual genes, but the other important thing is actually seeing genomes. And so like I mentioned, one of the big things that was out there was sequencing the Human Genome Project. Now, when I was going to graduate school, this is during the time of me going to undergrad and graduate school, they came out with this insane, like I said, the original, we're going to run gels and scan these things, we're going to put all these people on this, and they thought it was going to take about 25 years to get all the sequences because, again, we average, billions of copies of DNA. By the time we figure it all out, probably take us 20 to 25 years to do it. Well, with all the revolution and all the basically changes in technology in that, we were actually able to get it done within 13 years, which was a major driving force. So we actually had the first human genome sequence back in 2003. And this led to a lot of changes in the way that we do things. Genetic counseling. We can now look and screen people's genes to see if they have a mutation, if they're a carrier for something like sickle cell or factor V Leiden or something else. That may make your decision of whether or not to have kids, or if you are going to have a child that has one of these things that has a genetic abnormality, that maybe you want to say, do we want to have this child or not, or other things, or maybe we know what we're getting ourselves into in those things. So it's really changed those things. Like I said, it's changed ancestry. We can look and see ancestry out there and see who we're related to, go and doing those different things. and looking at a lot of different things that are out there. And so it's really opened the door to a lot more technologies that are out there, and all due to this. Now, since that time, they've been doing more and more genomes, human genomes, and that stuff. And so what they've found is, again, most of it, when they first did it, they sequenced about 92% of the genome. The last 8% was finally sequenced actually last year, which was pretty important because, again, it found some of the DNA that wasn't easily sequenced before. And again, finding new genes and also the ends of the genes and what those look like. And like I mentioned, really now the next project is getting multiple genomes, because not every human looks the same unless you're identical. Again, we're looking at what are differences in our genome and what it tells us about us. And so the Sanger Institute, which is over in Europe, they have actually sequenced now up to about, I believe it's over 240,000 human genomes. And so that's amazing. And so we can put these things in gigantic databases, look at these genomes, see what's similar, what's different, and it can lead to a lot of new things. And like I said, medicine's getting very individualized, and this is one of the ways that it is, by having these genomes to our availability and allowing us to see what these genes actually are. And so that's going to be really important. Other genomes that are very important are archaea bacteria, bacteria themselves. Like I mentioned, a lot of people use studies on bacteria, and this is, the estimated number 160,000. Like I said, bacteria have a very small genome, so sequencing those can be very easy, and you can see that a lot of those are done. And like I said, eukaryotes, much more difficult because you have multiple chromosomes in that, but still a good sizable number of eukaryotic genomes that have been sequenced to date. So why is this important? Again, sequencing, knowing what's out there, has led to new advancements. And one of the new advancements is actually being able to go in and change our genome. And so that's really the next advancement in editing our genome. And so this is something that you may hear of. And I get students every year asking me, what is this CRISPR thing I'm hearing about? I hear that this is really cool technology where they can edit the genome and do these things. Well, this is what it is. It's a very basic process that's actually done in bacteria. And it was discovered in bacteria back in the late 1980s, early 90s. And then it was then applied to eukaryotic chromosomes. And so back in the early 2000s, it's been applied, started to work. And then basically in the early 2010s, this is when this whole new technology started to take off, and using it in human genomes in this case. And that's why the two scientists that won the Nobel Prize in 2020 won it for this CRISPR, because they're using it and applying it in human genomes. And so what this is basically where you have a guide RNA, this CRISPR RNA, this cRNA, using this Cas9. And Cas9 is called essentially a protein that goes in and edits DNA. And so what's interesting is bacteria use this to remove bacterial viral DNA. Remember we talked about how I mentioned how sometimes DNA viruses can integrate their DNA in? Well, one of the ways bacteria can fight this off is they can find these pieces of viral DNA, and chop them out of there. And they use this Cas9 system to actually do this. And so it's kind of a unique little system that's found in bacteria and archaebacteria, but not found in eukaryotes. And so they can use the system to chop the DNA out using these specific RNAs. So what they've done now is use this in human genomes by using a specific RNA sequence that targets the DNA that you want to change, and then you can change these different ways. And so there's different ways to do this by actually activating mRNA, repressing RNA or DNA in this sense, or even visualizing, seeing where the gene is on the genome. And so you can do a lot of different things with it, and it's leading to a lot of new technologies. And so obviously, now this is getting more into the realm of playing God. So if you have a genetic mutation, you could potentially use the system to fix that genetic mutation so that you could alleviate certain conditions that that patient may have. And like I said, this is where that individualized medicine is coming about. And so I wanted to just show you some of the studies that CRISPR is actually being used. One of the big areas that it's being used is in blood disorders. And one of the biggest blood disorders is sickle cell anemia, where the misshapen cell causes major problems. So one of the things that we find is that if we could revert back to fetal hemoglobin, so the hemoglobin that babies are born with early on, that don't actually have the problem, that's one way to correct the issue, or potentially providing gene replacement of a normal hemoglobin gene. And so that's one of the ways that we do it. Another really interesting one is in cancer research and taking T cells and programming them to specific cancer cells. And so what we can do is make T cells recognize specific cancer targets that go in and destroy cancer cells. And so this would be a phenomenal way of destroying cancer without harming the body in other ways. And so the one issue that they're dealing with is how do we make it more broad, because right now they figured out very individual that we can take a person's T cells, reprogram and put them in. That's a hard process. If it would be much easier if we could just make a general pot of T cells that we could give to anyone at any time, fight the specific cancer and eliminate. And so that's the goal of the new next gen studies, is looking at how do we manipulate T cells so we can make a pot of T cells to give to anyone and make it less individualized in those cases there. Another one we're seeing is genetic blindness, where you're fixing, the cones and rods of people that have genetic mutations, diabetes type 1, to prevent, again, the cells of the pancreas to make insulin, again, and not get destroyed by the antibodies and those types of things out there. Chronic infection using bacteriophages that are programmed to destroy bacteria. So using using antibiotics, but now using viruses to actually kill the bacteria in the body, which don't harm people. Protein folding diseases, where proteins don't fold right, getting the right proteins in the right shape, and so they're working. Hypercholesteremia, so people that make hyper amounts of cholesterol instead of using like statin drugs or other types of drugs to lower the amount of cholesterol, again trying to manipulate the genes to make normal amounts of cholesterol in those cases there. And then HIV and AIDS, trying to remove the DNA out. So like I said, the Cas9 system is a bacteria's way of getting DNA out of the cell. Why can't we use that in human cells and being able to get the DNA virus out of the genome? And so this is another way of basically taking that DNA out of the viral DNA out of our cells and eliminating the threat altogether, because it hides. And that's one of the big problems with HIV, is that can hide and have this latent period for a number of years that are out there. So lots of cool things coming with these studies, and there's probably a lot more that I didn't mention here that are happening. So take a look, do a Google search on the Crispr studies, and I'm sure you could go down the rabbit hole and learn a lot about these different things. So this is just, again, touching the tip of the iceberg here with some of the different things that are being done. Now, like I mentioned, a lot of these early techniques are being brought to the elementary schools and high schools and everything else, and that's to pave the way for the future molecular biologists. And why do we need this? Well, to develop future techniques to work on these things. One of the things we're looking at is expression, and so using these gene chip arrays that have lots of genes on them and being able to compare disease to healthy. And so this basically takes a snapshot somewhere in the disease process where you can look at someone whose genes are being turned on in a disease state versus those that are healthy, and maybe saying, okay, what genes can we shut off to get a person back to healthy that's not found in the disease state? So that's one of the interesting techniques that are out there. Another one is, we know the DNA sequences, but what are the proteins that are made? And like I said, there's been about 20,000 genes that have been identified, and that's probably, again, that doesn't mean all the protein products that are identified, because there's splice variants and all these other things that are out there, but that just gives you the sheer number of just genes that are out there, or proteins that are being made. Trying to understand the proteomics is basically how do the proteins now function once you've made them. And so that's a whole nother area of study, looking at how either normal proteins or irregular proteins work, and why do they or don't they work in the sense of an organism. And so that's going to be another one of those things, and maybe working together cooperatively in those studies, looking at those as well. And then the last one that everyone wants to know about is these stem cells. You've heard a lot about these types of things and what goes on. Again, it was very controversial before, because we take them from fetuses, and some people would be like, nope, I'm not touching anything that's been from a fetus before. Nowadays, we can take fibroblast cells from people and use those to reconvert using a mixture of different enzymes in that, and convert cells back to the original stem cells. And this has revolutionized, again, this area where you can do things, all kinds of different things, in taking cells and growing new nerve cells, growing new heart cells, growing new skin cells, and you can just think of the different opportunities that you could use there. This picture down here is growing a bladder. So those that maybe have to get their bladder removed because of bladder cancer or other issues with their bladder, you could grow one up in a petri dish and give that person back. The other idea is, and this is a technology that we hope that's coming soon rather than later, is with transplants, in that people rely on people either providing that organ for, like a kidney or a piece of liver or something else, or potentially, if that person dies. And we don't want to see, we never want to hear about that, but they provide this life-saving benefit to someone else. Well, what if we don't have to worry about someone dying to give those organs, but you could make your own? One of the issues with transplants is the immune system. And so anytime you get a transplant, you're put on immune suppressors, because anytime you get someone else's organ, your body is going to recognize it as foreign and wants to get rid of it. No matter how well they try and match that organ, you want to be trying to be as successful as possible to match the thing so your immune system doesn't recognize it. Unfortunately, eventually your immune system it's going to cause rejection, and that's going to cause, failure of that organ. Well, what if we could make an organ that's you and built out of your tissue, so that your immune system says, yep, that's me, I'm not going to touch it, and leave it alone? And that's the idea of this transplant medicine using stem cells, is that can we build organs that we can now go and replace? And so now you could kind of think of it like an auto mechanic. You have bad brakes, you go into the auto mechanic and they can replace your brakes. Well, you go in and you have a bad heart or you have a bad lung. Well, we can grow that in the lab, replace that, and now you're functioning normally again. And so you can go on and keep replacing things as you go. Great technique if it works. Again, there's been stumbling blocks here and there, but they've moved forward. They're building hearts using cadaver hearts and things like this, using stem cells, and hopefully this technology advances to the stage where we get to that point where you can take these stem cells, repair nerve injuries, repair tissue damage, all these other things that are out there, and see if you can reestablish these things that happen. And so that would be wonderful. And then that would make life, go on much farther. And that if you could keep replacing things that start wearing out, the parts wear out, and you can replace them, and you could live potentially longer and longer and longer. And we may see people living to 150, 200, 300 years old based on just replacements alone. And so that could be a real interesting study in the future, and what happens. And so it wouldn't surprise me to start seeing these technologies happen in the near future very quickly, where we start to see these things happening very, very quickly in replacement of these organs. Okay, so just to kind of give you an idea of where we're going in the future and that stuff. And like I said, a lot of this has become because of this molecular biology that we learned in the past that we now can apply to the future and the future technologies. So like I said, it really all stemmed from that opening and closing of the DNA. And again, that was the basic principle of DNA. Once we understood the structure of DNA back in the 1950s and looking at the double helix and how that works with the hydrogen bonds, we could figure out how we could open and close it very easily. Then I mentioned the toolbox. Again, any good molecular biologist should understand these basic principles. And like I said, they're now becoming very standardized, that we're teaching them in the middle schools and high schools and even elementary schools, and how to cut DNA, how to glue it back together, how to measure the DNA using gel electrophoresis, copying the DNA so that you have enough of it to use over and over again, reading the DNA with being able to sequence it and do those things, and then finally editing the genome with Crispr and using those enzymes and being able to change, mutated DNA, getting target DNA to remove certain pieces of DNA, all those different things that can happen. And then, like I said, the future studies looking at expression of your DNA, protein structures and how that works with and plays with the rest of the organism, and then finally using those stem cells to build new things. And then, like I said, all this technology that we have today is based on the stuff that was used back in the 70s, 80s, and 90s that is now becoming commonplace. Okay, so I'll finish up with there. If you have any questions about any of this stuff, it's very exciting. I think, we're going to really see this change in medicine happen very, very quickly. You guys that are going to be at the front line are going to notice this faster than I am, and I'm always interested to hear new stories about what they do and how they do this stuff, especially in infectious disease, and how we're combating these things, especially to avoid resistance and all these other things that are out there. But I think it's going to be one of those things that are really going to change technology and how we treat patients as a whole. So if you have any questions, let me know. Otherwise, enjoy the stuff. I hope you kind of get some ideas out of it, get some ideas of where things are going, and maybe you've seen some of these things being used in a practical scale. And like I said, it's going to be more and more commonplace as we go along. So I'll leave you with that thought. Any questions, let me know. Otherwise, I'll see you in the next lecture. Talk to you then. Bye for now.